dua. Yang pertama nanti dari Prof. Watano itu akan memberikan terkait dengan apa kemarin tuh? Oke, bagaimana, kenapa kita harus mendefinisikan spesies? Nanti berdasarkan track record atau pengalaman risetnya dia akan menceritakan ke Anda semua, kita semua. Lalu Pak Asakawa akan menyampaikan risetnya terkait marine invertebrate ya. Kebetulan kami tahun depan merancang kegiatan lapangan. Jadi kami mengundang ada sekitar 20 mahasiswa Jepang yang akan datang ke Indonesia tahun depan. Kalian juga diundang untuk bergabung. Ya. Nanti kita akan field trip di Karimun Jawa 4 hari, terus kemudian ke Prokerto, terus kemudian ke Pantai Selatan, terus ada juga aktivitas presentasi dan diskusi bersama, terus kemudian ke Bogor, dan kemudian pulang. Dan itu tahun depan kita laksanakan di 2 minggu terakhir di bulan Agustus. Kalau Anda tertarik silahkan cek tanggalnya dan Anda bisa daftar. Baik, untuk kesempatan pertama kami memberikan kesempatan kepada Pak Dekan untuk memberikan sambutannya terkait dengan kegiatan ini. Waktu dan tempat kami persilahkan. Dekan, silakan.
Sini pesan sini aja ya? Apa di sana?
Terserah.
Ya udah. Saya baca aja deh. Terima kasih Pak Romi. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Selamat pagi. Ohayo gozaimasu. Distinguished guests, Professor Watano and Dr. Asakawa, welcome to Fakultas Biologi Universitas Sudirman Purwokerto. Ya, para wakil dekan, Pak Karsa, Pak Hendro, kemudian Pak Dr. Bayu Aji, Kepala Head of Research Center of Biodiversity and Evolution. Kemudian Pak Erwin tidak hadir, belum hadir, jadi para ketua bidang ada beberapa. Bapak Ibu dosen yang saya hormati, and all of our beloved students yang saya banggakan, alhamdulillah ya kita pada pagi hari ini insya Allah akan memperoleh banyak hal. Ya tadi disampaikan oleh Pak Karsa bahwa telah hadir di hadapan kita bersama Profesor Dr. Yasuyuki Watano. Ya, tadi insya Allah nanti akan memberikan satu materi. Tadi disampaikan bagaimana seharusnya kita mendefinisikan spesies atau spesies organisme ya. Kemudian nanti Dr. Takeshi Asakawa tadi juga disampaikan bahwa akan mencoba memperkaya kita dengan informasi keragaman invertebrata laut ya, yang ada di sekitar Jepang. Ya, ini jadi luar biasa. Good morning, ladies and gentlemen. In the name of Allah, clement and merciful, may His prayer and blessing be upon the Prophet and his followers. I would like to thank you all coming here, and I am sure we are all in healthy and fine this morning. It was great for me and Universitas Jenderal Sudirman University to host distinguished lecturers from several countries, including from Japan. Many thanks to Watano Sensei and Asakawa Sensei for giving our students the chance to learn from you and for establishing a collaboration program between Jenderal Sudirman University and Chiba University. Hopefully, the summer course on tropical biodiversity that will be offered the next year will be very beneficial for us in strengthening the biology study program. I also want to express my gratitude to Dr. Romanus and Dr. Erwin, the Director of the Office for International Relations, for the support of this program. I hope our collaboration will earn several benefits, not only for our students, but also our network, Jenderal Sudirman University and Chiba University, or between Indonesia and Japan in more advance. Prof. Watano and Asakawa Sensei, have a nice day here in Purwokerto with very good weather. Inshallah, very nice city and also nice people. Yeah. Dan perlu kami sampaikan juga untuk teman-teman ini luar biasa sekali lagi. Jadi mohon nanti bisa dirancang, tadi ditawarkan oleh Pak Romi, ya. Agustus, insya Allah nanti kita ada satu kegiatan bersama dan informasinya juga ada teman-teman mahasiswa dari Jepang, ya Pak. Ada mahasiswa dari Jepang juga hadir sekitar 10 atau mungkin 15 bisa bersama-sama dengan teman-teman semuanya. Aktivitasnya sudah dirancang oleh Pak Erwin dan Pak Romi dan Pak Erwin, ya. Dan mudah-mudahan ini menjadi satu momen pengayaan kita semua. Ya. Again, I would to say thanks to all of you. Arigato gozaimasu. Selamat pagi, mohon maaf atas kekurangannya. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Pak Dekan, for your welcoming speech. And now I will invite Pak Bayu. Pak Bayu nanti akan jadi moderator karena kebetulan dia profesornya. Jadi kita selalu senang mengundang Pak Bayu. Pak Bayu kebetulan juga dosen apa namanya ya Pak Indro programnya? Kampus mengajar ya, dari BRIN mengajar di sini ada 2 dosen ya kalau nggak salah ya. Pak Dekan matur nuwun. Nanti kita lanjut dengan ini, monggo berkenan untuk kalau masih stay di sini silahkan. Kami mengundang Pak Bayu ke depan untuk jadi memperkenalkan profesornya dulu ya, silahkan ya. Hadir di tempat ini tidak terlalu banyak, so not so many here, but actually we have 120 plus online. So join with us online 120 participants. So yeah, maybe more than that.
And then YouTube also.
128, yeah, also YouTube. So please, Pak Bayu, time all yours. Before that, is it possible to get your presentation? So because we deliver the presentation in Zoom, so I would like to have your copy of the material, PowerPoint or something. So we deliver in that computer.
Copy.
It is. PPT no file ga copyしてZoom にshare します。もし
Do you have any file for your presentation?
でもZoom で
Maybe just
online.
copy.
How to bring? Yeah.
Pakai itu aja USB.
Do you have a flash disk?
Saya ada flash disk.
Zoom nggak ada yang sekarang. Posta, sedikit-sedikit saya bisa bahasa Jepang karena saya pernah studi 6 tahun di Jepang bersama dengan Pak Bayu, pergi dan pulangnya sama satu pesawat juga, dan dia orang hebat, saya biasa-biasa. Sebelum kita lanjutkan dengan perkuliahan, kalau ada hal yang terkait mau ditanyakan terkait dengan program-program dari BRIN boleh. Sudah ada yang masuk ke periode apa? MBKM, terus riset misalnya? 2019 ya. Kalau yang riset itu S1 barista itu bantuannya 3 juta S1. S2 6 juta, S3 9 juta. Dan semua kegiatan bisa dilaksanakan di sana gratis. Ya. Kemudian ada teaching assistant juga, terus kemudian ada apa lagi Bapak Bayu? MBKM magang.
Degree by research.
Terus setelah lulus bisa degree by research, lanjut beasiswa dari BRIN di perguruan tinggi yang bekerja sama dengan BRIN. Utamanya saat ini PTNBH seperti ITB, LIPI, IPB, Kuliah.
Ya luar negeri juga ada. Malaysia, Taiwan, Australia, Inggris sudah join dengan program. Ya.
Oke, itu dibiayai oleh Pak Bayu. Karena yang tanda tangan dia.
No.
Jadi kalau tertarik silahkan daftar sampai S3 lalu balik lagi ke sini jadi dosennya. Silakan Pak Bayu waktu ini.
Oke, thank you. Terima kasih. Ohayo gozaimasu. Sensei. Hello. Selamat pagi. Good morning, ladies. So much ladies ya. It's okay. Thank you very much, Pak Dekan, Pak Romi, and also all my teacher in this faculty. So let me introduce myself first. My name is Bayu Aji, actually, and then I'm graduated from this faculty in 1997. Maybe you're not born yet. Yeah, still, okay, no problem. Good luck. Okay, I was studying plant taxonomy at this undergraduates, the only student who learned plant taxonomy in this faculty. So I learned about the pterophyte ferns here, and then I'm joined to botanic garden LIPI at that time, and I have a chance also to continue my master's and my PhD in Watano Sensei Laboratory, also doing research in ferns, mostly in speciation and also in molecular systematic of the fern. And now I work in as a director for research center for biosystematic and evolutions. So this is the center for all taxonomies in Indonesia. So for plant, animal, also in microbe, you can, of course, you are a good talent to welcome you join later on in our institute if you're interested in taxonomy. So we have more than 130 researchers in my center. Actually, that is for later on. You can ask what Pak Romi said. And now let me introduce our speaker here, Professor Yasuyuki Watano is a professor in plant taxonomy and then evolutions. He studied for more than maybe 3 decades in taxonomy. He is graduated from University of Tokyo, and then become a professor in Chiba University. So when I was in Chiba in 2002, he became a professor there. Then I joined proudly joined to his laboratory for 6 years. 6 years. So today he will present also about the species and speciation. What this is the main area of the evolution. Very interesting field. We talk about biodiversity, but biodiversity is product of evolution. How species become exist on the earth. So this is always a very good discussion. Jadi saya juga pernah memberikan kuliah di sini tentang spesies dan speciation ya. Jadi bagaimana spesies itu bisa muncul di muka bumi ini. Dan ini adalah merupakan bahasan yang sangat menarik dalam dunia evolusi. Dan ini silakan bagi teman-teman, ini biologi tentunya akan sangat relevan dengan apa yang akan Anda pelajari nanti. Evolusi tidak hanya bicara tentang morfologi, but evolution also can detect from the molecular side. Jadi we're talking about the molecular evolutions. Ready? Sudah? Okay. Now time is yours, Professor.
Yeah.
And I will. You can present there. Oh, okay. You can also Yeah. Maybe you can stand here and then. Yeah.
Hi, selamat pagi. Hi. Thank you for Bai-san. Thank you for introduction. So thank you for giving me a chance to give a talk here. I came here to develop a student exchange program between General Unsoed University and Chiba University, Japan. So in Chiba University, we have 5 campuses, which is located in Chiba Prefecture, here and here. Chiba Prefecture is near to Tokyo Station and Narita Airport. Narita Airport is a gateway of the Japan. So I would like to see some of you in future in Chiba University. I'm expecting you. Yeah. Next slide, please. How to do that? Oh, right. Okay. My presenter's title is How Should We Define Species of Organism? This is my illustration drawing by my student. Okay. Hi. So can you appear this? Thank you. Difficult. Okay, no problem. Yeah. If you ask what my specialty is, so. Okay. So, okay, yeah. If you ask me what my specialty is, I will say I am a plant taxonomist. So, what is the job of a taxonomist? Okay. So, well, it's a job to recognize the basic unit of biodiversity species and to resolving evolutionary relationship among species and giving taxonomic systems. For example, grouping closely related species into the same genus and grouping further closely related genera into a family. Okay? Hi. This photograph shown here is a wetland farm. I give name is Scatopteris gaudichaudi variety vulgaris. Okay, so let's take this as an example and explain about the scientific name. Okay, what is a scientific name? Each organism have a name such as human and dog. So, but if you go to a different country, and the name may be different. So this is a problem. So scientists give a universal name for each of species given in Latin. This is a scientific name. Okay. In this case, Ceratoptes is a genus name, and Gaudichaudi is a given to distinguish this species from other species of the same genus. Okay. This looks as a family name and given name for us. But the scientific name should be must be only one in the world. Hi. One of the joy of the taxonomist is you can put your own name to the scientific name. Yeah. In this case, this is my name. Hi. The way of thinking about species has changed greatly before and after Darwin. Do you know? Maybe you know Darwin. Darwin is famous for his theory of evolution. Okay. Before Darwin, it was believed that species were created by God and are immutable. But after Darwin, on the other hand, Darwin argued that species change over time. He believed that all species diversified with modification from a common ancestor. This is what Darwin wrote down in his note. This may be the first phylogenetic tree. Oh. The reason why we can recognize species in this world is because world of organisms are discontinuous, okay? Our world is species is discontinuous, so we can recognize species. Here is a question. If species were created by God and immutable, then discontinuity between species is likely, okay? But if organisms change, as Darwin said, the world of organisms may be in a world in which species change from here to here. So maybe species change continuously from one species to another. So human, Shiba dog, okay? While species are discontinuous, if it's strange when you think about it, okay? And to explain this answer, let's discuss the definition of species. Biological species concept, in abbreviation BSC, is now the most widely used definition of species. In this definition, species are distinguished by the presence or absence of reproductive isolation. Within the same species, organisms can interbreed and exchange genes. Between different species, organisms cannot exchange genes because they are reproductively isolated. This figure shows there are many reproductive isolation. Here, color of the soil is different. This means these two species growing in different habitat, okay? If they grow in different environment, they will not meet. So they will not interbreed. This is called habitat isolation. Here, only upper plant have a flower. This means the breeding system is different between two. This is called temporal isolation. In the end, these two plants make a seed and hybrid seeds. These hybrid may be maybe inviable or sterile. This is called post-mating isolation, okay? There are many steps of gate for reproduction. So in fact, biological species concept, BSC, can explain the discontinuity of the biological world. Within the species, genetic exchange through crossing homogenizes species and give a cohesion force, so united into one species. When two species are isolated by reproductive isolation, the accumulation mutation occurs and the difference grows with time. Okay. Oh, so can you see this here? Oh, thank you very much. Mm. Hi. This small plant, this small plant is a plant I showed earlier. Characteris Gaudichari variety vulgaris. I separated this species from this large one. As you see, the size difference is great, but there are intermediate forms. So before my study, all types are grouped in one species, Characteris electronics. I found out hybrid between two species are two species are genetically differentiated. I conducted the artificial crossing test, and the hybrid between two have produced irregular spurs, as you can see. Maybe this black spur is empty, and has no DNA. So this means the two species are reproductively isolated. Online, okay. So difficult to so. So this reproduction, seed production with pollination is called agamospermy. Okay. The product, yeah, this is how to say agamospermy. Okay. Swarmous means chaos. How to say chaos? Yeah. Yeah, this is the reproduction of seeds without pollination. Okay. Do you have any question? You can stop by talking every time, okay? So now this is the central theme of this lecture. A second example of the drawbacks of BSC is a species that cross with each other and exchange genes. The two spine species shown here, Pinus pumila and Pinus parviflora, are the main character of this lecture. Pinus pumila is a shrub growing in the alpine zones, and Pinus parviflora is a high tree growing in the mountain zone. They grow. Sorry. Hi. They grow different environment, but this reproductive isolation is not complete. They make hybrids in some mountains and exchange genes with each other. To show that three forms of the two species are genetically fixed, please see this photograph taken in Mount Apo in Hokkaido, where we can see that the two species grow side by side. This creeping tree is a Pinus pumila, and this is a sapling of a Pinus parviflora. So this grow to high tree, but they are still creeping in adult, okay? In Japan, mountain zone is defined as the altitude range between altitude range of the summer green deciduous forest, such as Calgus. Okay? Subalpine zone is the range of conifer forest, such as Abies or Tsuga. And alpine zone is the mountaintop region above timber line. This pink shaded line is a distribution range of Pinus pumila from Russia to Japan. Japan corresponding to the southern tip of the distribution of this species. So in Japan, distribution of Pinus pumila is restricted to this mountaintop. So temperature decrease around the altitude. So this is very cold place, and so this plant is restricted to the alpine zone in Japan. Okay. Pinus parviflora is mostly endemic to Japan and distributed from mountain zone to subalpine zones. So hybrids between two species is occasionally you can look this in inter-equatorial zones. We call here to this zone of the hybrid. It's a hybrid zone. Okay, please remember. So this is relatively difficult. So consideration. Okay. And there are many hybrid species. Let me explain the merit of choosing pine species as materials. Plant have three kinds of genome. One is nuclear genomes, and one is mitochondrial genomes, and one is plasmid genomes. In most plants, both cytoplasmic genomes of the mitochondria and plasmid is inherited maternally. So only maternal genome is transferred to the offsprings. However, in pines, mitochondrial genome is maternal, but plasmid genome is paternal. Okay. So mitochondrial DNA and plasmid DNA have different mode of inheritance. So they can be transferred independently, even in crosses between the two pine species. Okay. Here I explain that although plants can't move, gene transfer occurs using two media. One is seed and one is pollen. Genes can move by seeds and pollen. Okay. Seed is a capsule of young plants. So all three genomes, nuclear, mitochondria, plasmid, is carried by seed. But pollen is difficult to say, so male gamete for it. So don't carry mitochondrial DNA, only nuclear and plasmid. Since pines wind pollinated, so pollen grains are dispersed long distance by the wind. Considering this, the moving of mitochondrial DNA, which is not carried by pollen, maybe moving ability of mitochondrial DNA is much smaller than seed. Okay, seed is just drop here, but pollen can fly to long distance. Okay, in this way, a difference in mode of inheritance also lead to difference in dispersal ability, moving abilities. Although there are two types of cytoplasmic genomes, there may be they may also be difference in how genes move between species. Take a care look at the following observations. The first thing we should do is to develop molecular markers to know the origins of DNA of the collected samples in hybrid zones. The rest here is the band pattern of the electrophoresis of plasmid DNA. This is conducted by the PCR SCP method. Bison also using this method. So I will not explain the detail over here, but PCR SCP is a method that can detect nucleotide mutations in PCR products with very high sensitivities. So, the left lane, this is Pin's pumila, and this is a parviflora. You can see a band pattern is different. Okay, so these samples W13 to W21. This is collected from hybrid zones. You can see only W14 have a pumila type DNA, and other samples have a parviflora type DNAs. The right is an example of the DNA marker with mitochondrial DNAs. PCR products of the mitochondrial DNA, NAD1 was electrophoresed on the agar scale. You can see that the size is different between Pin's pumila and Pin's parviflora. This is a size marker, so you can see that pumila is a smaller DNA than parviflora. Okay, so you can distinguish these two species easily on agar scale. Hi. The first analysis was conducted in the Taniga Mountains on the border of Gunma and Niigata Prefecture. I started climbing here, so climb along this river and here, Shimizu Pass hut. I stay overnight here. So next morning, I walked along this ridge line and down to the Doai. So this is this white dot is the site of the collection of my sample. Okay. Okay. Let's start with the plastid DNA. この circle is a plastid DNA and red is a pumila type and blue is a parviflora type. You can see most of the plants are parviflora types. The only pumila type sample is found around the peak of the Mount Asahidake and the peak of the this mountain. Next, this triangle is mitochondrial DNAs. So here is the low elevation ranges. The both parasitic and mitochondria is blue. This means that this is a pure type of parviflora. Okay. Beyond here, we can see most samples have parviflora type plastid and pumila type mitochondrial DNAs. The combination of blue and red. Okay, most of the samples has a combination of the blue and red. I organized this result by arranging the samples. Here, samples are arranged from low altitude to the high altitude. Here is mountain foot and this mountain top. You can so here we also examine the morphological identification. So you can see morphology transform from paraflora type plants identified paraflora, so intermediate type and to mountain top type. Okay. So in this plastic DNA form column, we can see paraflora plants have a paraflora type plastic. Intermediate plants also paraflora plants type DNA. And some of the Pumila plants also have paraflora type DNA. So paraflora type DNA is introgressed to the Pumila. So this direction from paraflora to Pumila. Okay. Here, next, this column, you can see the Pumila type plants, morphologically identified as plants, Pumila have a Pumila type mitochondrial DNA. And intermediate plants also have a Pumila type mitochondria. And some of the plants identified as a paraflora also have the Pumila type DNA. So mitochondrial DNA flow this directions from Pumila to paraflora. So mitochondria is introduced to this directions. Therefore, gene flow, so as a result, in these hybrid zones, cytoplasmic genomes with paraflora type DNA and paraflora type plastic DNA and Pumila type mitochondrial DNA is dominated. Okay. So this slide is also difficult to understand. So explain in details. Instead of using morphology, here we use the hybrid index. Okay. Hybrid index shows a quantity of the mixture of nuclear genomes. How much percentage of the Pumila genome and how much percentage of the paraflora is included in one individual. So I quantified by hybrid index. For example, 1 means 100% Pumila individuals, and 0 means 100% paraflora individual. So this plant is 90-100 Pumila and 10% paraflora. Okay. Oh, sorry. This white graph is altitude of the collection of the samples. Here, this steep line is means the gap of distribution in this elevation zone from 1,300 to 500. Okay. And the black graph is the hybrid index, the indication of the mixture of the two species. It is noting that although the hybrid index, hi. Yeah.
Yeah, okay. The male parents and the female parents is interchange. It will result in the same things in the mitochondrial DNA patterns and the plastic DNA patterns. Are they the same if the male parents and the female parents are changeable?
So can you explain this?
Yeah. Which one is the male parent and which one is the female parents in the hybrid here? Is Pumila
So
or
I'm sorry. These plants are hermaphrodite. So make both sexual hermaphrodite. Okay.
Hermaphrodites?
So
Okay.
female flower have female flower and male flower. So I can understand the balance.
So okay.
Mm.
So it doesn't matter whether which one is the pollen source and which one is the
So every nature have a pollen source and seed source. Okay.
okay.
Yeah.
Yeah. Okay. Thank you.
Hi. So Hamnix suddenly increase this zone also after the gap of elevation. So, but here is the top of the mountain, but this plant is not completely pumila. These plants have 30% of the peripheral genomes. So there are no pure pumila plants in this mountain. Okay.
Excuse me, Professor. If we look for the picture, there is very different in morphology between Pumila and Pavipora. And can you mention about the hybrid morphology? It is different, or how it look like?
Sorry. So
Thank you.
I identify by using the needle. Pine has a needle, so I cut the anatomical character. I use the anatomical characters. So this is a more resin canals. So resin canal is called this type is pins pumila, and this outer zone is a palpebral type. And also another character is also exist. So, but more characters is also so. Yeah. So pumila cleavage shrabian. So this is but so most intermediate plants have some shrubby. So not a high tree, but very difficult to using this cleavage shrub or trees difficult to use for identifications. So sometimes high wind zones, tree forms is called creeping. So difficult to use this habit to use distinguished species. So I use anatomical characters. Okay. So intermediate plants I identified here is plants not identified to even to be our pergola. Okay. Hi. So do you understand? In this mountain, genetic mixing is like this. And so here is a pure polyflora, but here is no pure pumila in even the top of the mountains. We can know this type of data by using molecular markers. In addition to Taninga Mountains, we conduct the same analysis for Mount Higashiazuma and Mount Zao. At the top of Mount Higashiazuma, you can see a pink zone. Pink zone means both cytoplasmic genome is pumila. Okay. Under this pink zone, we can see only green zones. Green zone means plastic is palpiflora, but mitochondria is pumila. In this mountain, all plants have this type, so green zones. Overall, the heterospecific combination plus the mitochondrial DNA, so different combination is only found in this collection. You can see, we can expect the plastid pumila and mitochondrial paliflora, but these plants is not found. only this type of heterospecies combinations. This is a rule, okay? This is very interesting observations. We can explain this pattern by assuming that the direction of gene flow is fixed. Crops plus the DNA flow from mountain foot to top, but mitochondrial gene flow is different under the directions. This is a rule. The difference among mountain may be created because the levels of introgression varies from mountain to mountains. Okay? If a gene flow of the plus DNA is limited, we can expect pink zones. Okay? If gene flow of the mitochondrial DNA is limited, we can expect blue zones. Okay. So now there are no blue zones in Mount Higashiyama and Zao, no blue zones. This means that all samples have pulmonary type mitochondrial DNAs. Okay. The next slide shows the geographical distribution of these extensive mitochondrial DNA introgressions. Here I collected palmiflora plants and examined the genotype of the mitochondrial DNAs. This pie graph shows the frequency of the pulmonary type mitochondria and peripheral type mitochondria. Just in the southern part of the old mountain, you can see all red graphs are concentrated. Red graph is concentrated here, these regions. Okay. In this zone, mitochondrial DNA derived from Pinus pumila completely replaced the native mitochondria. So I collected peripheral plants, but in this region, all plants have a mitochondrial DNA of the all plants are pumila type. So this means suggesting that in this zone, native DNA replaced by the alien mitochondrial DNAs. This phenomenon is sometimes called the cytoplasmic capture. Okay. This figure on the right is examining hybridinix analysis of this Mount Zao in the center of the hybridization. In this blue zone, this is a lower elevation and plant sample is a tree. But here is a high altitude zone, and so plants collected a creeping shrub. But in both, this tree form is here and this creeping form is here. So all of the plants are hybrid indexes are mostly even. So this is surprising that it is interesting that although both are genetically 50% mixed, they are divided into all tree and shrub in outer morphologies. Okay. Okay, higher. So do you know who won this year's Nobel Prize in Physiology and Medicine? The prize was awarded to Svante Pääbo for his discovery concerning the genomes of extinct hominins and human evolution. He determined the genomic sequence of Neanderthals, the closest extinct species to modern humans, for the first time. He also found traces of past interspecies hybridization in the human genomes. Dr. Pääbo and his group shows that ancient human hybridized with closely related species such as Neanderthals and Denisovans. Here is the lineage of the human lineage and Neanderthal and the Denisovan diverged around this 600 to 500 kilo years ago. And human go out Africa and move to Eurasia. In Eurasia, a human meet Neanderthal and Denisovans, and so hybridization may occur around here. Okay. Genetic exchange between species would be a common biological phenomenon. This is the take-home message of this lecture. Okay. So I don't give an answer to the curious result of my study. One is why were gene flow plus and mitochondrial DNA fixed in one direction. So my hypothesis is this: only F1 hybrid could be formed only in the direction of the Pumila as maternal and Pariflora as a paternal prime. So I tested this hypothesis by crossing species, but I failed. So I don't confirm in this, but maybe I believe in this maybe the explanation of this phenomenon. So this is a very good, very large question. Why are species maintained for exchanging genes? Maybe this answer is a natural selection as a species. So in BSC, gene flow is acting as the coefficient of the species, but evolutionary force is also including natural selection and genetic drift. So in this case, natural selection is a coefficient of, I think. Okay. Thank you very much. I finished the lecture.
Okay, very thank you, Professor. Jadi bahasan tadi cukup sangat menarik ya buat kita semua bahwa driving force untuk evolusi, untuk pembentukan jenis, salah satunya yang paling banyak melalui gene exchange, jadi pertukaran gen, dan itu difasilitasi dalam proses yang disebut dengan hibridisasi. Nah, di sini beberapa contoh tadi diberikan ya. Pertama dari tumbuhan paku itu Mizuarabiですよね。Mizuarabi. Jadi Ceratopteris thalictorides. Kebetulan saya mempelajari Ceratopteris thalictorides untuk independent origin of the cryptic species in Ceratopteris thalictorides. Kemudian ada contoh juga dari spesies bunga ya. Lupa. Dan yang terakhir yang banyak dibahas tadi tentang pinus ya, pinus. The most special in the pinus adalah pada umumnya kloroplas DNA itu transfernya maternally, right? But in pinus is special, this is paternal. Paternal. So ini lebih memudahkan kita untuk menganalisis mana orang tuanya ya. Jadi mana asalnya dari mana ininya induk jantan dan induk betinanya di situ, dan di sini sangat spesial bahwa hanya one direction, but hybridization only one direction, not two direction. Jadi Pinus pumila itu betinanya, Pinus parviflora jantannya. Tapi hibrid ya, tapi tidak berlaku hibrid itu tidak pernah ditemukan parviflora itu adalah betinanya dan Pinus pumiflora itu jantannya. Jadi only one directions. That is one direction is still in question. Why? Usually we can make it two direction ya. Jadi bisa aja bolak-balik. Iya. Oke. Jadi hybrid zone bisa terjadi karena perbedaan altitude tadi ya, dari yang pumila ada di puncak kemudian parviflora ada di bawah, tapi di tengah-tengah itu ada hybrid zone tadi ditemukan. Nah, hybrid itu tidak selalu kemudian morphologically sharing. Nah, jadi tidak selalu kemudian sudah menampakkan karakter morfologi intermediate. Dan tadi diterangkan bahwa ini tidak ditemukan di hybrid di pinus tadi. Jadi mereka masih menunjukkan sifat masing-masing. Nah, itu tapi secara anatomi tadi sudah bisa kita lihat bahwa sudah ada intermediate feature in hybrid F1-nya sudah ada. Hanya tadi secara morfologi, secara kasat mata masih belum bisa dilihat hybrid ini ada karakter intermediate ya, karakter pertengahan. Kan biasanya kalau ini panjang, ini pendek, tengah-tengahnya jadi sedang kayak gitu loh. Nah, itu belum terlihat, jadi masih hidden, dan ada kekuatan sendiri yang dalam natural selection. Jadi kira-kira seperti itu, jadi masih banyak question ya, so many question need to be answered by all of you, and of course this will be enforced by molecular method. Question? Is any question from Zoom? Can you? I have to read it. The question. Sorry. Where is the question? Okay.
First question is, so how to use SHP, not SSR, ISSR, ISR. Yeah, so SHP is a very easy. So by distinguishing SSR, we must use sequencer. But SSR is just electrophoresis under the constant conditions of temperature. So very easy to use SHP. So if you use difference by using SHP, you can determine sequence. So very effective to correct which sample should be sequenced. Okay. So next question is, how do you determine if a sample is morphologically intermediate? Just macroscopic or have to get microscopic? Yeah, I used only the anatomic character. So this is a microscopic character. Okay. So this character may be not affected by the environment. So I used this anatomical character. So next is, do environmental factors that habitat altitude affect change in species when no other species in the genus are found? Can I understand the question? Last question. The environmental factors that the habitat altitude affect change in species which no other.
Jadi
this is a question of the environmental global warming. No? I'm sorry, so I can't understand.
In all species, that altitude and environment change. So his study is now in pinus, not another species. Maybe if study abroad in other species, maybe we'll find maybe different case.
So, I thinking that our two pine species, Pumilia and Parviflora, maybe have more chance in
another this
in
different
the difference of environment.
different species.
So this environment change may affect the frequency of hybridization, I think. So I think I suppose his question is maybe so.
Yeah. So kenapa tadi ya SSCP ya, simple sequence conformations polymorphism. SSCP itu sederhana dari hanya electrophoresis, tapi juga cukup sensitif. Jadi satu perbedaan satu nukleotid itu bisa dengan mudah dibedakan bandnya, ya. Dan itu pada suhu ruangan. Jadi tidak perlu mesin yang terlalu canggih. Hanya kita butuh gel sama electrophoresis, kemudian kita soin, kemudian kita bisa lihat tadi bendingnya. Even one sequence difference, the band will be spirited. Even the sensitive, quite sensitive. Hanya saja kelemahannya dia hanya fragmen yang bisa dianalisis itu sekitar 300-an. Ya, 300 base pair. Istilahnya tidak terlalu panjang. Tapi bisa membedakan dengan cukup sensitif. Jadi dibandingkan tadi dengan ISSR apa kan harus pakai sequencer. Take time, take money. Kayak gitu. Another questions?
So he may be interested in the chemotaxonomy. I'm studying the cryptic speciation with the ferns. It's difficult to distinguish morphologically. So I think chemotaxonomy is very good method to detect the cryptic species. In my study of the aspirin, so in the previous study, some types of chemotype is described in the aspirin normale. So I examine these plants by using molecular marker. So most of the chemotype is correspond to the different species I found. So I think chemotyping is a very useful method to distinguish a cryptic speciation, I think. Okay.
Ya, oke. Selain chemotaxonomy juga mungkin yang protein ya. So like isozyme, allozyme. Jadi
maybe chemotaxonomist is maybe flavonoid.
Flavonoid.
Secondary compound.
Yes, secondary compound. Yes.
Okay, I want to ask a question. Please, I want to know if there are any difficulties in biological species concept. Any difficulties in the application?
Any difficulty, if any difficulty to direct the biological species concept in your case?
So these two are exchanging genes. So if we apply this VSC to this group, Pumilia Parviflora is included in the same species. But these species are morphologically and also ecologically different. So yes, grouping one species is nonsense, I think. So this is not applied to the interbreeding species, so I think.
Okay, so dalam hal banyak in the case we apply what species concept is applied in the case is sometimes depend on the case. In this case, a special biological concept can apply, but morphological species concept you cannot apply. After you can find morphological differences between. So in this concept, we have to consider in any case. Different case maybe will be applied different concept. Right?
regarding the band you show us around 2000 something around 2300 or 2300 between Pumila and another species. Can you explain to us what exactly different it is, or do you found some kind of a sequence that
Oh, so I used 10 genetic loci of the nuclear markers. So I examine the likelihood of the each species. So this is means a hybrid mix. Okay.
okay.
So next.
Yes, that's one.
Next one. Is
So you show us that
next one.
Yeah, 2322 and 2027. And can you yeah. Yes. Yeah.
This is sorry. So you answered this.
What the exact difference of the sequence?
I examined the sequence, and so this any1 gene have a very long introns. So many in the insertion or deletion exists. This is the difference of the size of the PCR product. Yeah.
This is not related about some kind of amino acid, or only just interspace or something.
Yeah, most of the PCR contains an internal regions, and so there is few coding regions. So difference is about very.
So, jadi ini adalah itu adalah produk PCR ya.
Ya.
Jadi produk PCR kita lihat bahwa itu panjangnya berbeda-beda. Itu dikarenakan adanya intron. Jadi intron yang berbeda itu akan melihat panjang pendeknya nanti sequence gitu.
Jadi yang membedakan antara antara satu spesies A dengan spesies B bukan kepada gen fungsionalnya dalam hal ini,
ya,
tapi pada intronnya
ya,
saja.
Untuk PCR kan karena kita tidak melihat sequence ya. Kalau PCR kan kita hanya melihat panjang atau pendeknya ke hasil PCR-nya. Nah, ya, sorry. Jadi I just explain what you said in bahasa Indonesia. Jadi PCR tadi itu sekitar ada yang 2000 berapa ya? Itu yang membedakan tuh intronnya.
Oke.
Intronnya.
I also use this sequence to construct a phylogeny.
Yeah.
So this large difference is corresponding to the different lineages. Yeah.
So.
Targeted sequence, a particular sequence. Do you use a particular primers or just the whole mitochondrial DNA and the whole?
Just these regions, about 2000 base pairs.
Yeah.
So what primers do you use?
Universal primer is developed for coniferous regions.
Okay. Is there another question? One more question maybe from student. Maybe you will be Watano Sensei. If you asking, maybe he will interesting to you whether you can be his student or not. Bring one to Chiba. You can choose one of the as your student later. So, jadi ada just expand to encourage them to study taxonomy, and then also taxonomy is not about herbarium, it's not about preserved material in the museum. So taxonomy is not all about genome. Now, so di era sekarang itu yang disebut dengan era nomik ya. Jadi genomic, proteomik, jadi omik seperti itu. Jadi teman-teman nanti sudah, you should be ready for the next decade that we should not talk about the genome. Tidak hanya sekedar gen, particular gen, tapi mungkin whole genome. Jadi di fasilitas kami di BRIN itu sudah memfasilitasi jika Anda mau men-sequence whole genome sequencing. Jadi kami punya next generation sequencing Illumina, dan kami juga punya ONT, Oxford Nanopore, ya. Jadi Oxford Nanopore Technology. Yeah, of course. If you have some collaboration with us. Ya, jadi not too much, you talk too much. Kami juga nanti di sana punya berbagai fasilitas ya untuk herbarium terbesar di Asia Tenggara, juga museum zoologi terbesar juga. Jadi memfasilitasi bagi teman-teman, student untuk bisa belajar, you can learn taxonomy from morphology to genome. Yeah. Yeah, we must do everything. So chemotaxonomy, isozyme, protein is old school, right? Even we can still understand and usable, but that is old-fashioned, old school. Anyway, is there any question? No? Close, then thank you. Give applause. Unsur also with BRIN research center or our institute. We already have a letter of agreement between Fakultas Biologi and Research Center for Biosystematic and Evolution. So let's use this chance to get research collaboration between us. Thank you, and then I will pass the time to Mas Roni.
Oke, applause for Pak Bayu dan Pak Watano. Baik, seperti disampaikan Pak Bayu tadi bahwa kami sudah punya MOU dengan BRIN, terutama di Pusat Riset Biodiversitas dan Evolusi. Kami sedang membuat pusat kolaborasi riset antara Unsud dan BRIN, khususnya dalam biodiversitas ya. Kami merancang waktu itu biodiversitas pesisir ya. Tapi mungkin bisa kita kembangkan biodiversitas laut, pesisir, dan pegunungan. Baik, next lecture will be delivered by Dr. Asakawa, Takeshi Asakawa. I will be the chair here. Kemarin dengan Dr. Asakawa kami sudah jalan-jalan ke Karimun Jawa selama 3 hari di sana. Dan ada big prize, ada hadiah besar di sana ketika kami pertama kali ketemu hiu paus, whale shark, close encounter, jaraknya cuma 1 meter gitu ya. Dan itu memang ekspektasi kami datang di sana di sekitar bulan November-Desember karena mereka itu juga ada meeting ya. Hiu paus itu ada meeting di sana, meeting tahunan, annual meeting untuk mating. Annual meeting untuk mating di sekitar situ, daerah itu memang tidak banyak yang tahu, hanya sedikit yang tahu, maka kita ke sana, siapa tahu awal Desember masih ada, dan ternyata masih ada 1 yang tersisa. Mungkin di tempat lain masih ada juga. Langsung Dr. Asakawa juga dari Chiba University, dari lab yang sama juga. Dia mempelajari banyak hal, mulai dari tumbuhan, dan sekarang lebih fokus pada invertebrata, karena sebenarnya bidangnya adalah phylogeny ya, phylogenetics. Oke, Dr. Asakawa, time all yours.
Thank you, Lon-san. And thank you very much for inviting me to Indonesia and giving a chance to lecture today. Okay. I was paleobotanist first, and then I was botanist and studied mangrove plants with Arvin-san, and plant-animal interaction. Today, I am a marine zoologist, and I'd like to introduce diversity of some marine invertebrates around Japan. I hope this lecture will help to promote collaboration and exchange program and your education. So, this is a photo of nude branches from my Instagram account. So, next one, please. First, biodiversity refers to the variety and variability of species and of interaction of them. The biodiversity recognized in 3 levels: genetic diversity, species diversity, and ecosystem diversity. Today's lecture, I introduce mainly species diversity. Please. According to UNCP, United Nations Environment Program, the number of species is estimated to be between 3 millions and 111 million. The reason for the wide range in numbers is that many species are expected to be undescribed. Species diversity is much greater than currently recognized. I say this hidden species diversity. This is chart of species number. Most diversity on Earth is arthropods. Next one, please. The cryptic species. Cryptic species refers to the species which are difficult to distinguish morphologically, but are genetically distinct. This is an example of cryptic species of certain nudibranch, Petroeolidia. For example, Petroeolidia yantiana is the only known species, but genetic study, they were divided into 2 species. And they are difficult to distinguish by external morphology. Such cryptic species would be the cause of hidden biodiversity. Next one, please. And this is a famous example of cryptic species in your country. Orangutan is distributed in Sumatra and Kalimantan. And Kalimantan population and Sumatra population is divided in 2 species at 2001. And in this paper, one of Sumatra population here is quite different in genetically quite different. Sumatra, main population Sumatra is here, and small population here is genetically different. So they describe this population as Fungo tapanuliensis. So you know well, I think. Next one, please. So in the history of Earth, the Earth appeared 4.6 billion years ago. And organisms appear about 3.8 billion years ago. This is the known oldest organisms, fossil. They are in rock of stromatolite. This rock is called stromatolite. And black lines contain carbon. And the source of carbon is a small organism like this. And then the first organism is prokaryote. And then eukaryotes appear and multicellular eukaryotes appear. And the animals appeared about 600 million years ago. And then extinction and organization of various taxa had made today's biodiversity. Next one, please. This shows big five mass extinction. So many taxa had originated and been extinct during the history of life. The extinction rate over the last 500 million years, this red line indicate extinction rate. So we can recognize five peaks of extinction rate. So we call this big five mass extinction. The biggest was number 3 at the end of Paleozoic. And the second biggest was at the end of Mesozoic. The end of Mesozoic is famous for the extinction of dinosaurs. Next, please. So we were in the 6th mass extinction in progress. This graph shows extinction rate at this time, each of this time. At the end of Cretaceous, the time of dinosaur extinction was estimated at 0.001 species per year. And increase extinction rate. And at 1975, it was estimated about 1,000 species per year. So we are in the 6th mass extinction. Next one, please. So local extinction refers to a situation in which a species or population no longer exists within a certain location. This figure shows local extinction. So species A has three populations. One of these population 2 disappear. This is local extinction of species A. But if the extinct population is cryptic species, it would be global extinction. So right side, if population 2 is cryptic species B, so this is not local extinction, but global extinction of species B. Next one, please. So revealing the hidden species diversity is important, I think. Now we are in the age of mass extinction. So cryptic or undiscovered species may extinct before we realize them. So it is important to reveal the hidden species diversity of life to prevent the extinction of undescribed species. Now, the analysis of gene sequence has made it easier to find cryptic species. DNA barcoding analysis and molecular species delimitation analysis are useful tools that can be used to identify species by comparing DNA sequence and to reveal inter or intraspecific diversity. Next one, please. So DNA barcoding is a technologies and the DNA sequence databases for identifying species simply by sequencing. It is applied to these areas. Next one, please. And the molecular species delimitation analysis can also be used to determine how much sequence difference constitute different species. This is a principle of ABGD, automatic barcode gap discovery. So sequence of many samples are compared with each other. And this is value means sequence difference number, and the count is how much and here you can see boundary of value. And you can divide into two species. Next one, please. I would like to show my study of hidden species diversity of Limnoria. Limnoria is a marine isopod feeding on usually wood. But I study Limnoria feeding on brown algae. So Limnoria feeding on brown algae were collected from several kinds of algae, Eisenia, Sargassum, and Padina. They feed on mainly these three algae. The DNA barcoding and the phylogenetic analysis can reveal hidden diversity of Limnoria. Next one, please. This is a phylogenetic tree of Limnoria feeding on brown algae. They are divided to 3 or 4 monophyletic group. And yellow group feed on Eisenia. Orange group feed on Eisenia and Laminaria. And purple group feed on Padina. And blue group feed on Sargassum. Each monophyletic group and the host seaweed were associated. Very interesting. Next one, please. So one of the recognized monophyletic group was the known species, Limnoria nagatai. But the other monophyletic groups are not undescribed species. So we described the 2 new species, Limnoria fulca and Limnoria aspera. They are very similar in morphology, but we can distinguish them by dactyls of secondary unguis of Paleopod 1. Here. This dactyls is different by species. So next one, please. We also perform species delimitation analysis and recognized 8 species. 3 species for Limnoria nagatai, 3 species for Limnoria fulca, and 2 species for Limnoria aspera. And this is a distribution area of each taxonomic unit. So 3 or 2 species occur sympatrically, and side of Pacific Ocean, usually 3 species occur sympatrically, and 2 species sympatrically Sea of Japan side. Taxonomical unit 3.1 is okay. Next one, please. So I'd like to introduce accumulation of DNA barcodes. DNA barcode data are accumulated at board systems. This is URL. The DNA barcode databases were created in the CO1 region for animals and in the MATK and RBCL regions for plants. This is a start page of board systems, and here is a tab of identification. So if you click here, next please. This is the identification page. You can choose database and paste sequence here and submit. Next one, please. If similar sequences are available in the database, the species can be easily identified. If similar sequence is present, you can see scientific name, result of identification here. and taxonomic information here. But similar sequence is not present. You can't identify. So it is important to accumulate significant sequence data in the database. Thus, it is significant to clarify the sequences of species that have not yet been registered. Next one, please. So I'm collaborating with Sea Slug World on the DNA barcoding project for nude branches and sea hares around Japan. So Sea Slug World is a photo database of nude branches and sea hares, mainly around Japan. But 763 entries from Indonesia are also registered in this site, Sea Slug World. Next one, please. The accumulation of sequence data of nude branches that forms the basis of DNA barcode databases is progressing. However, only a few sequence data on Japanese nude branches and sea hares have been accumulated. Thus, we have started to analyze DNA sequence data of nude branches and sea hares around Japan by Sea Slug World barcoding project. As a result, we are beginning to obtain interesting results, such as the discovery of cryptic species, undescribed species, and misidentification, etc. Today, I would like to introduce some of the results as follows. Next, please. This is DNA barcoding study of nude branches of Indonesia by Duharamawan et al. They collected nude branches widely in Indonesia. Red is the sample point. And identified them by DNA barcoding. Next one, please. Next, please. DNA barcoding identified 18 species out of 51 samples analyzed. This is a figure of identified species. And I interested in Chromodoris. 4th species was identified in Chromodoris from a broad area of Indonesia. And this study showed that molecular analysis can identify nude branches to species. Species level identification is possible. Next, please. I promoted similar studies on the diversity and undescribed species of Chromodoris in Hachijo Island. Hachijo Island is far south from Tokyo, small island. I collected from one diving site in Hachijo Island here. And 13 samples were sequenced and identified by DNA barcoding. Next one, please. This is the result. DNA barcoding identified 9 species out of 13 specimens analyzed. So this is a phylogenetic tree over Chromodoris. And red letter means my sample from Japan. So of these, number 6, number 8, and number 11 is new sequences. And maybe undescribed species. Number 11 is morphologically identified as Chromodoris rochi, very similar to Chromodoris rochi. But the sequence is quite different, number 11 here. So it may be cryptic species. And number 6 here is this one. It is named in Japanese. Japanese name is Kongasri umiushi, Kongasri nudie branch. And number 8 is this one, Honnoriro nudie branch in Japanese. So they are morphologically predicted new species, but is not described yet. Here I can show they are independent and the new, they have new sequence and they are independent species. Next one, please. Next is example of micromero. Micromero undatus was the only valid species until recent. But they collected from all over the world and make DNA barcoding and phylogenetic analysis. The species delimitation analysis resulted in 4 species. So Micromero undatus divided into 4 species. And new species Micromero barbarae was described. The Micromero barbarae is a specimen from Hachijo Island. This is not my work. Blue letter means citation. Right. Next one, please. Next. This is a result of prior paper. Micromero divided into 4 species. And Micromero undatus is distributed in Atlantic Ocean. And Guamensis in tropical area of Pacific. Barbarae is temperate area of Pacific. No. Barbarae distribute wide in the Pacific. No. Scriptus is distributed widely in the Pacific region. And Barbarae is temperate of Pacific. So next, please. Next. So I collected some Micromero specimens from Hachijo Island and performed DNA barcoding. DNA barcoding identified Micromerovarbarae and Micromeroguamensis. Here is Micromerovarbarae, and specimen 1 is Micromeroguamensis. And I found Micromeroguamensis look like this, with blue edge on the ventral foot, blue edge. This may be characteristic of Micromeroguamensis. You have this type of Micromero in Indonesia, blue type. Next one, please. This is example of Coryphelina. Coryphelina rubrolineata is a nudibranch with a conspicuous purple line on its back. Purple line here. Very characteristic nudibranch. The Ekimova et al. Perform DNA barcoding and phylogenetic analysis and species delimitation analysis, and show many cryptic species is present. Right side is a photo of this kind of nudibranch from Japan. Next one, please. The molecular species delimitation analysis results show that Coryphelina rubrolineata complex is divided into 8 species, and they describe 4 new species. Next one. I collected this kind of nudibranch and performed DNA barcoding. This is the result. I found Coryphelina rotos. This is originally described from Japan. And I found this one, Coryphelina pannae. And specimen 2 and 3 is a new sequence. So it may be undescribed species 2 and 3. Next example is Durabrifera of Appreciated. Valdez et al. Valdez et al. Collected Durabrifera durabrifera. Durabrifera was only valid species. They collected Durabrifera from all over the world and perform DNA barcoding analysis. And they divided them into 6 species. 6 species. Durabrifera distribute tropical in the Pacific. Very broad distribution area. And others, red one is Atlantic Ocean. And the blue, this species is distributed in temperate Pacific of Southern Hemisphere. And this species is distributed in East Pacific, coast of South America. So I collected Durabrifera specimens and performed DNA barcoding analysis. I found Durabrifera. It is known to distribute Pacific Ocean. And I found two undescribed species. Specimen 2 is monophyletic to Durabrifera acifera, which is distributed in Atlantic Ocean. And specimen 3, very close to Durabrifera edmundsi, which is also distributed in Atlantic Ocean. And I found this number 4 specimen that is very different from other sea hares, and does not belong to any genera of the Apricidae. Maybe another genus. Next one, please. And this is an example of Meribe. This specimen is very small and was considered to the juvenile of Meribe viridis, this species. You have Meribe viridis in Indonesia. The DNA barcoding analysis indicates that Meribe sp, this one, is has a different sequence and maybe undescribed species. Next one, please. So DNA barcoding is useful for recognition of juveniles. This pink small sea hare feeding on red algae. The sequence of this sea hare is identical to that of Aplysia chloride. You have Aplysia chloride in Indonesia. So this small pink sea hare may be the juvenile of Aplysia chloride, this one. So in Japan, this sea hare is identified as Aplysia juliana sometimes, but it must be juvenile of this one. The body color may change with the shift in food algae. Juvenile feed on red algae and adult feed on brown algae. Next is example of juvenile sometimes recognized as separate species. This is Keratosoma bicolor. And this is Keratosoma trilobatum. C1 sequence of this Keratosoma bicolor is almost identical to that of Keratosoma trilobatum. So, Keratosoma bicolor may be juvenile of Keratosoma trilobatum. So, this name should be the synonym of Keratosoma trilobatum. By famous nudibranch researcher Baba, he recognized two types of Keratosoma trilobatum, A type and B type. And he also described Keratosoma bicolor. So this is phylogenetic tree of this, these three samples. The A type and B type are different, quite a different sequence, and they should be separate species. And B type almost identical to Keratosoma bicolor. And this name should be synonym. And thus, A type new scientific name should be applied for A type, this type. So you have A type of Keratosoma in Indonesia. So this is a summary. DNA barcoding analysis of sea hares and nudibranches of Japanese specimens is ongoing, and Sea Slug World DNA barcoding project. We are beginning to obtain interesting results such as the discovery of cryptic species, undescribed species, and misidentification, etc. Even narrow and thin sampling revealed a variety of interesting facts. More sampling would reveal the diversity of nudibranches and sea hares around Japan. Next one, please. So in Indonesia is one of the hot spots of sea hares and nudibranches. Tramban in Bali is a famous diving site with diverse nudibranches among Japanese divers. By DNA barcoding analysis, you will be able to discover the hidden diversity of nudibranches and sea hares in Indonesia. Okay. So I hope this lecture help promote our collaboration and your exchanging program. Thank you. Yeah, next one. Thank you for your attention.
Thank you very much, Asakawa Sensei. Jadi menarik sekali apa yang tadi kita lihat, apa yang dipresentasikan itu betapa pentingnya untuk mengeksplor hidden diversity. Ya. Jadi Indonesia ini tempatnya karena nggak ada yang meneliti. Gitu ya. Pak Bayu punya pasukan berapa? 100 berapa Pak Bayu? 137 peneliti di BRIN. Belum lagi peneliti di perguruan tinggi, tapi itu masih kurang karena penelitian biodiversitas di Indonesia masih relatif kurang. Ya. Jadi Anda terbuka sekali untuk meneliti. Ya. Apalagi tadi yang disampaikan Prof. Asakawa, Dr. Asakawa tadi bahwa dari nudibran saja banyak sekali ya. Tadi disampaikan ada banyak, bahkan di Jepang yang penelitian diversitasnya sangat ekstensif pun masih banyak ditemukan spesies baru, apalagi di Indonesia. Tadi disampaikan kalau yang nudibran siapa tadi Pak Darmawan ya tadi Pak Darwan itu mungkin cuma sendirian ya. Kalau di perguruan tinggi masih sangat jarang atau bahkan tidak ada yang meneliti tentang nudibranch. Kalau kalian terjun di satu tempat seperti kemarin kita di Karimun Jawa, diam di satu tempat, di Lamun saja misalnya itu Anda belum tentu langsung lihat ya, karena di daun itu biasanya ukurannya kecil, ada yang cuma 1 senti, ada yang kurang dari 1 senti, dan itu kalian harus diam satu tempat untuk bisa melihat. Itu memang perlu effort untuk melihat di satu lokasi diversitas tersebut ya. Hiddennya itu bukan karena tersembunyi, tetapi memang banyak jenis yang belum terdeskripsikan. Terus tadi yang menarik adalah bahwa apa tadi yang makan rumput laut itu dari makanannya aja bisa menuju kepada pemisahan clade ya, yang nanti bisa menuju pada spesiasi mungkin ya. Kalau dari kalian misalnya ada yang suka nasi goreng, ada yang mie goreng itu makan itu terus nanti bisa spesiasi itu. Nanti jenis mulutnya beda ya, ada yang menghisap mie-nya kan dihisap tuh. Kalau nasi kan dipatuk. Ada pertanyaan silahkan. Ya silahkan. Marcel.
Okay. Okay. Thank you very much for the opportunity given to me. My name is Aditya Marcel Junior, a student majoring in biology at General Sudirman University, and at advance, arigatou gozaimasu and congratulations to Asakawa Sensei on the excellent presentation. Allow me to ask. I have planned to do similar research to yours for my final research. The topic is species diversity and abundance, especially the sun dollar. The topic is the sun dollar. Yeah, the echinoderm, the sun dollar species in Karimun Jawa. The topic is the species diversity and abundance. In that study, I will calculate the diversity and the abundance of species based on morphological characteristic directly on the site. Before you said that there are cryptic species that can only be distinguished through the molecular methods, right? So my question is, how can I really distinguish one species to another species based on morphological character directly on the site, due the limitation of bringing all the available sample to the lab, because there's so much individual that I should take. So do you have any suggestion to solve the problem? Thank you, Bifar.
So, genetic species, genetic species delimitation is you can do by several methods of species delimitation analysis. We usually use consensus of several methods. But in the case of Limnoria, species delimitation analysis identify 8 species, but I can't distinguish by morphology. I can distinguish only 3 types. So description is now restricted to 3 species. So you can distinguish genetically by species delimitation analysis, and you can use consensus of several methods. But it is difficult to describe them or not. You can just separate by genetic diversity.
Okay, arigato sensei.
Question? No? Someone is raise hand. Ya. Pak Joko langsung aja open mic. Tolong dibuka mic-nya, saya tutup. Ya. Sudah. Silakan Pak Joko.
Thank you very much for the time. I have a very basic question. So if there is a different identification result, can you hear me?
Yes, sure.
Yeah. If there is a different identification result between morphological approach and molecular approach, which one should we trust? Because this is the case in my laboratory recently. So a gastropod species was identified by an expert, and then she also did the DNA barcoding. But when the result coming up, it shows completely different species, like freshwater and marine species. So in this case, which result should we trust? Thank you very much.
Pak Joko bisa diulangi lagi.
Okay. My question is very simple. If there is a different identification result between morphological approach and molecular approach, which one should we trust? Because this is the case in my laboratory. So a gastropod species was identified, by an expert. And she also did, besides morphological approach, she also did DNA barcoding. But when the sequence result coming up, it shows completely different results. For example, by using morphological approach, it's identified as species A from freshwater, but the DNA barcoding shows that it's a marine species. So in this case, which one should we trust? Or is there something wrong with the technique of maybe DNA extraction, or I don't know, because I'm still learning about it. Thank you, Professor.
Okay. In my case, I collect as many as nudibranchs around Japan and sequence first. And I hope to find some interesting taxa. And then I will study such taxa by deep collection and morphological analysis. So one of the merit of DNA barcoding is easy to identify by not specialist. So if you are specialist of some taxa, DNA barcoding and morphological study is both important, and you can do that. But if you are not specialist of certain taxon, or you are not a taxonomist, you can do DNA barcoding first. Okay.
Yeah, but the question is, it's already identified morphologically, and the genetic sequence come up. When there is disagreement between morphology and genetic, how you solve the problem, and which one should we trust?
It was identified by an expert, a taxonomist.
Already identified by the expert.
Already. So in the case of nudibranch, genetic identification first, and morphological difference is variety in the species. It's just only the case of nudibranch.
Itu Pak Joko ya. Jadi kalau dia hanya bisa jawab kasusnya nudibran. Kalau dia trust genetik pertama, yang kedua adalah morfologi sebagai variasi.
Ini kasusnya karena beda sekali, jadi morfologi itu menghasilkan freshwater species, dengan barcoding itu marine species, gitu. Jadi totally different, atau mungkin metode kita yang kurang, ada sesuatu yang something wrong di mungkin DNA ekstraksinya atau bagaimana, mungkin.
Tapi sampelnya diambil dari laut atau dari tawar?
Freshwater.
Hah?
Freshwater, tawar.
Freshwater, tapi keluarnya marine.
Iya.
Wah. Ya silakan, Pak.
Oke, thank you.
Oke, Pak Joko mungkin ada di sini kalau biasanya kalau di molekuler hal itu bisa terjadi, tapi harus dilakukan pengulangan, Pak.
Oke.
Nah, biasanya kalau pengulangan kedua kok dengan yang pertama itu sama, berarti ya itu memang terjadi dalam tanda kutip memang itu unik gitu. Mungkin bisa jadi spesies yang baru gitu ya. Ya, intinya memang untuk memastikan bahwa kesalahannya ada di mana. Apakah kalau di morfologi itu kan sesuatu yang bisa dilihat. Tapi kalau di molekuler kan mutlak itu kerja-kerja enzim. Jadi perlu ada kalau result pertama error, jadi perlu ada pengulangan lagi untuk memastikan bahwa result yang pertama dengan yang kedua itu sama gitu. Biasanya seperti itu, Pak. Sehingga bisa jadi kalau di kasus saya mensequence untuk satu spesies bisa sampai 3 kali. Karena tidak yakin bahwa sekuen yang pertama itu memang itu. Sehingga memastikan sampai 3 kali. Itu seperti itu, Pak.
Mungkin, kalau sudah diulang hasilnya konsisten, berarti mungkin kita lebih percaya ke pendekatan molekuler mungkin ya, asumsi
ya, kalau
saya.
itu konsisten, justru yang dipercaya malah yang molekuler.
Oke, clear, thank you.
You're welcome.
Baik, ada pertanyaan lain? Kalian kok nggak nanya bagaimana saya terlibat dalam proyekmu, Pak, gitu kan? Bagaimana saya bisa ikut ke Chiba misalnya? Nggak ada yang tertarik dengan itu, nudibranch? Kami memang mencari kerjasama ya. Dia sudah bisa menerima untuk proyek kolaborasi. Kemudian dia memang mau eksplor di Karimun Jawa, di perairan-perairan lain dan butuh kolaborator. Dan tentu di sini ada Pak Bayu juga yang punya izinnya, yang tanda tangan izin peneliti asing tuh Pak Bayu itu yang tanda tangan. Kalau nggak pakai akunnya dia nggak ada yang diizinkan peneliti asing. Jadi silahkan kalau Anda tertarik, Anda bisa kontak langsung, bisa kontak via saya bagaimana untuk kerjasama atau terlibat dalam penelitian ini. So Marcel is my student under my supervision for the $10. So we will go to Karimun Jawa also to collect $10. So if we identify by morphology, and then we also continue with genetic. So hopefully he can send the sample to you for the genetics. Hopefully, so we can collaborate then. Begitu ya. Ada pertanyaan lain? Ya. Silakan. Mic-nya di mana tadi ya?
Thank you very much for your lecture. Actually, I would like to know about the barcoding itself. In the database, it is only use one barcode method like CO1, or use another method like maybe for another, like maybe another housekeeping gene.
Yes. This is the first stage of my work.
Okay.
Just barcoding of CO1 region. And if I found interesting taxa, then for phylogenetic study, we need at least 3 regions of
3 barcoding
DNA. So CO1 and 16S and histone 3.
Okay.
So
Arigato.
I found interesting taxon. So I will perform further study of phylogenetic study and taxonomical study, including description of new species.
Thank you. So you go first in the CO1, and if you found something different, then go deeply in another barcode.
Yes, yes.
Correct.
I am planning to barcode all species of noisy branches in Japan.
Oke. Arigato.
Baik, saya kira itu ya. Ada pertanyaan lain? Menarik karena tadi disebutkan dia menunjukkan bold ya, bold system ya. Ada juga blast system, biasanya kalau kita pakai blast. Tapi kalau yang bold system biasanya sudah diidentifikasi oleh taksonomis, yang bold system. Tapi beberapa kali saya coba antara bold dan blast, kok lebih dipercaya yang blast gitu ya. Diidentifikasi saya paling tidak. Do you have any idea what's different between blast system and bold system? Bold and blast.
You can get almost same result by bold systems and blast. So difference is the database. Some sequence in bold system is not registered in NCBI, and such sequence cannot search by blast.
Oke, thank you. Ada pertanyaan lain? Pak Endro, Pak Dekan, Pak Karsa, Agus. Jika tidak ada, maka kita berikan aplaus untuk Pak Asakawa. Thank you so much. So that's conclude our lectures here. We have 2 lectures, and I think for the closing remark, Pak Erwin, bisa kasih closing remark untuk kerjasama ini. Ini Pak Erwin, Direktur Aero, jadi saya kasih kesempatan. Silakan.
Ya, Assalamualaikum warahmatullahi wabarakatuh. First, I would like to thanks Dr. Asakawa and also Professor Watano to give a lecture to our students and also to our college via online, via Zooms. Thank you very much. And then also thank you very much, Pak Dekan, and Vice Dean for the academic, Vice Dean for the student and alumni affairs, our college docent, and also all of your our students who take class in Biodiversity and Evolution. It's lecture by Pak Romi. Thank you very much, dan semoga ini menjadi awal untuk bisa membuka kesempatan ya. Di samping membuka ilmu pengetahuan, membuka kesempatan untuk melangkah ke yang lebih jauh. Semoga melangkahnya bisa sampai ke Jepang, ya, karena banyak yang menarik di sana, ya. Tidak hanya ilmu pengetahuannya, teknologinya, tapi juga budaya dan etos kerjanya yang luar biasa. Again, thank you very much for all of us, and thank you Pak Romi for your guidings for these sessions. So, and also thank you very much my friends, teman satu angkatan. Beliau sudah jadi kepala pusat riset, luar biasa. Dan kalau ya biasanya kalau anak kos zaman dulu ya, main sesama kos itu main kartu gitu ya. Tapi tidak pakai uang. Dan juga terima kasih yang sudah bergabung di Zoom sampai tadi menyentuh 160 lebih. Terima kasih sekali dan semoga kesempatan ini bisa berulang di semester yang berikutnya. Hopefully you could possible to give lecture for the next semester, even offline or online via Zoom. Thank you very much. Assalamualaikum warahmatullahi wabarakatuh.
Thank you very much, Pak Erwin. So for the last session, we will, as token of our gratitude for your lecture here, we will give you a certificate. Could you come forward here, Prof. Watano, Prof. Asakawa here? We will give you a certificate and souvenir that you already given lecture here. Ya, terima kasih banyak Pak Dekan. Dengan demikian berakhir sudah kuliah umum ini. Saya sebagai host mohon maaf jika ada kekurangan. Terima kasih banyak kepada seluruh mahasiswa yang hadir. Marilah kita selalu semangat untuk belajar, untuk melakukan riset, untuk menemukan hal-hal baru dan semoga masa depan kalian semua cerah. Ya. Waalaikumsalam warahmatullahi wabarakatuh.